Okay, so we just covered in the last lecture about correlation and descriptive research methods. Now we're going to cover experimental research methods. Now, this is really important to think about in terms of what experiments can do and allow us to do. And so this is going to be just sort of a quick run through of what we're able to do with experiments. So experimental methods are the only research method that allows us to say causality. So we can say that A causes B, there's a cause and effect relationship between two variables or two factors because we have manipulated something. If we just look at something and don't manipulate anything, then we can't say that A causes B. But if we can actually get in and do an actual experiment where we have our variables that we manipulate and change on purpose in a systematic way, then we can, of course, say causality. So here we have two types of variables. Now remember, your variable is the thing that you're interested, the thing that you measure, the thing that you're paying attention to. It's the thing that can change. So we have two major types of variables that we talk about here in experimental methods. One is the independent variable, which is independent of everything. That is the thing that you're going to manipulate. It is the one thing you're going to change. The thing that changes in response to that, okay, is your dependent variable. That is dependent upon your independent variable. Sometimes you'll see these are actually broken down into IV and DV, so you can actually see them. A lot of times they're just abbreviated, but your independent variable is independent of everything. It's the factor that's independent of everything, and it is the thing that you are changing. The dependent variable is dependent upon that and is the thing that is changed. So if you run a study, for example, and you want to see if, let's say I have a class, and I want that class, I want half of the people to drink caffeine, and I want the other half to not drink caffeine. And I want everybody to do a chapter 1 test in psychology. And I want to make a comparison of the people who drank caffeine and the people who didn't drink caffeine. And I want to see who does better. Same test, one big group of students, same lectures, same everything. So the only thing that I change in this entire experiment is who gets the caffeine and who doesn't get the caffeine. So my independent variable, the variable that is manipulated, the one that changes, that I change specifically, is caffeine. Do you get it or do you not? And the thing that is dependent upon that is your test score. So I'm going to test your test score, and I want to see if that's going to change based off of what I manipulated. Okay, so I'm going to use this example. So remember that we have a hypothesis, and this is our question that we've asked, a prediction that we're going to test. Okay, so it's our educated guess. So I might ask essentially the question about, does caffeine impact test scores? And so my hypothesis might be based off of other people's research that caffeine will in fact improve test scores. So a good hypothesis tells us direction. It tells us, is it going to improve or is it going to get worse? And it has the factors involved. So my hypothesis might be to test if caffeine improves test scores. The main effect is going to be any change in those test scores that we can absolutely and directly attribute to our independent variable. So when we look at test scores, we are looking for those test scores to change, and that's going to be our main effect. The independent variable, this is what we manipulate. It's also sometimes referred to as a treatment variable, depending on the kind of study that you're running. So in this case, our independent variable is exposure to caffeine. Our dependent variable is what we're measuring and what is going to be influenced or dependent upon that independent variable. And so this is going to be your test scores. Any other extraneous variables are anything that we can't attribute, that we can't control, okay? Or that we can determine to affect our actual results. We also call these confounds, okay? And so these extraneous variables need to be corrected so that it doesn't mess up our results. So one confounding variable or extraneous variable could be that you walk into the room already have had caffeine and maybe you're in the non-caffeine group. Well, that's a problem because that means that that's going to affect, your test score. And so you would need to either try to control for that or remove you from the study, or you can statistically try to control it in some fancy statistics. So you might have a particular rule to control this extraneous variable that nobody gets caffeine before they walk through the door. That way, those who don't get caffeine truly do not have caffeine. And those who do get caffeine, of course, get an absolute amount that they're going to get and not more than that. Now, of course, you also have an extraneous variable of, well, what if you usually have caffeine and you don't get it? That could actually make you do worse because now you're going through withdrawal. So there are a lot of things to consider with experimental work, and you have to make sure that you've thought about all of them that you can manage. So you have an experimental group, and these are all the people who are exposed to the conditions. Okay, so in this case, we would have the experimental group of being the group of students who took caffeine. And then we would also have what we refer to as a control group, and that group gets nothing. That group does not get any manipulation. And in this case, it would be the group that did not get caffeine. So we'd have an experimental group who gets the condition, the experimental condition, and get the caffeine. And then we have a control group, which is not on here as a term, but these are the group that gets no kind of change whatsoever. With random assignment, we want to make sure that every single person has an equal chance so that we don't accidentally bias our results. We don't want to put everybody who consumes caffeine in one group and everyone who doesn't consume caffeine in another group, because that can actually mess up our data. We want it to be spread equally across all groups if it's going to be a random assignment. And we want to make sure that everyone has an equal chance to be randomly put into either group. Now, of course, not every study does random assignments. But hopefully the idea is that any differences or biases can be spread out across everyone. So if you have a big enough group, it should work. You can also have a single blind or double blind study. A single blind study is where the researcher is aware of everything. They know what's going on. They know what treatment you're getting. They know what the manipulation is, and they know what group gets what. But the participants don't know that. The subjects don't know it. Now, the term subject is usually referred to in psychology as an animal, and participant is usually referred to as a human. But we've made some changes recently in terms of the American Psychological Association that now it's kind of gone back and forth. It used to be that subjects could be people and participants were also people, and subjects could also be used as animals, the term used for animals. It's gone back and forth, but really today you can use them interchangeably. I would say 5 years ago, it was a big hubbub that you only use participants as people and subjects is the word used for animals. But now you see it kind of used both. Now, a double-blind study, this or double-blind technique is where you will see that neither the participant nor the researcher knows what condition they're in. Okay, so single blind, the researchers know that information, but not the participants. Double blind, nobody knows. And that's to reduce bias. We don't want the researchers to expect the participants to behave a certain way because they know that that participant is getting a particular treatment. So somebody in the group actually knows what's going on, and they actually have it coded so that you can match up the participant in the end and know who goes to what group. Okay. Now, a few sources of bias. Of course, you can have that a person thinks that something's going to change, and so they end up seeing a change. Okay, so we see a change in the dependent variable simply because the person is expecting a change, and that's an expectancy effect. We can also see demand characteristics, which is a bias that the researcher actually communicates to a person. They think they're getting a particular treatment, so they're expecting them to behave a certain way. For example, let's say a researcher knows one group is getting alcohol and another group is getting fake alcohol. And then the researcher might expect one group to act drunk and give cues off to that group. And if they don't know if they're getting alcohol or not, they may just simply react to what the researcher is expecting. And these demand characteristics can, of course, mess up your study. A placebo is something that you get that's like a sugar pill. It's fake. It has no effect on you. And it's just to level the playing field so that you think that you might be getting something or you don't know what you're getting. We can also see placebos take forms other than pills. For example, in treatments like we've talked about transcranial magnetic stimulation or TMS. It's a wand that goes over your head that's magnetic and can change, for example, can alter, make you remember better. It can also be a treatment for bipolar or depression. We actually see that you can do placebo that way by waving a fake wand and the person hears clicks and thinks that it's working. So this is just essentially a fake substance or treatment just to make sure that everyone thinks that they're getting the same thing. And then, of course, a placebo effect, which is an expectancy effect, is when you think you're getting the real drug and you actually change based off of what you expect is going to happen. And so that can be caused by thinking that you're getting the treatment, and that can be strong enough that it could actually make you think that it's working, and that can change your results. Now, we do have limitations of experiments. One thing is that, it's great that it's controlled, but sometimes it's actually too controlled. And so if you have too strict of conditions in the lab, this can make it unrealistic, and it doesn't actually relate to real behavior because it's so ultra controlled. Also, there's ethical considerations, and we're going to talk about ethics here in just a second. And we have to really always consider the fact that we have to make sure that the study is safe for our participants and actually is relevant to the kind of behavior that we would expect in real life. And sometimes if we want a really lifelike situation, we can't reproduce that simply because it would be unethical. So if you wanted to see how people behaved in an earthquake, we can't make an earthquake happen. If you want to see how people react to a burning building, we can't set a building on fire and see how that works. Those are all considered extremely unethical, and so something that we actually can't do. What we do find is that we can actually get more valid insight simply from naturalistic observation and combined with naturalistic or natural experiments. But again, there are where we can actually observe like a naturally occurring event. We have to, of course, wait for that event or condition to happen. We can't sort of recreate that. But that can be very difficult to do, even though it does produce us sometimes a lot better insight. Now, when considering ethics in experimental work, of course, we're going to talk. If you take social psychology, and when we get to the social psychology chapter, we're going to talk a little bit more about the experiment right here. That's a picture of what we call the Milgram experiments. And we'll talk about if that was ethical or not, and can it be repeated. But if you take social psychology, we spend a lot more time on that. It's very interesting understanding sort of how people behave in certain situations. Now, one thing that's very important to understand in research is that it's, every single person who's doing anything in research, in psychological research in the U.S., has to go by the ethical guidelines that the American Psychological Association developed. Now, it doesn't matter if you're an undergraduate, it doesn't matter if you don't have a degree, it doesn't matter if you have a PhD or an MD. If you're doing psychological research, then you have to follow these ethical guidelines. And one really important thing is to consider the fact that you, of course, if you're in an institution doing research, that you're absolutely going to have an ethics board who is going to monitor to make sure that you're treating your participants ethically, and to make sure that you are not hurting anybody. So before any study can be done on a human, we have the IRB, or Institutional Review Board, that's made up of a group of individuals. It's an ethics board. It can be teachers, it can be lawyers, it can be psychologists, researchers, it can be a preacher. They're made up of several different types of people, those with research background and those without, and they make sure that the research, that the risk and the benefit ratio is proper. So you want to make sure that the benefit outweighs the risk. So, it might be a very dangerous study, but if the benefit is really, really high, then they may let you do it. Anything that you do in your study has to be approved, and you can't make any changes without approval first. So even if you change one word on a form to get somebody's consent to approve to do the study, even if you want to change a word, you got to go back through the ethics board, and they have to make sure that it's ethical. So the 5 key provisions that the APA has for human research is, number 1, that the person has been informed of the study, and that they give their consent, that they are, in fact, they want to do the study, and that is completely voluntary, that they're not being coerced in any way, whether it's through money, whether it's through power, and someone, like, I can't have one of my students be a subject or a participant and threaten them that I'm going to fail them if they don't participate. That would actually be coercion. They need to make sure that they can quit at any point in the study. That is something that is very important. You could be all the way through, 99.9% through the study, and if you throw your hands up and say, I'm done, then you have to be allowed to leave, and you are not to be coerced to finish the study. Number 2, students are very vulnerable because, that the person running the experiment could be their teacher, it could be their advisor, it could have something to do with their grades or their graduation. And so students can be used as research participants, but they must be told that, of course, that they are completely voluntary, and that they do not have to do the experiment to get a grade. If you receive extra credit, for example, for doing an experiment, you have to be given an alternative so that you do not have to actually take part in a research experiment. The third one is use of deception. So a lot of people say, oh, well, you're not allowed to lie to people. You're not allowed to deceive them. If there is absolutely no other way around lying to a participant, in the sense that you will ruin your experiment if you tell them the purpose behind it. If you have justification, then you can absolutely deceive participants. If the whole point of the study is to see their reaction to something, and if you tell them in advance it'll ruin the experiment, then you can deceive them. But it's the most minimal amount of deception, and you have to have a justification. You can't just lie to people and say, oh, that's just, oops, okay, we'll lie to you and we have no reason. You have to have a justification, and then when you're done with the study, you have to do something called debriefing them, which is you tell them what the lie was, why you had to lie, and of course apologize for lying, and make sure that they understand, why it had to be done. The fourth one is confidentiality. No matter what you take from a participant, whether it's personal information, scores on a test, scores on a measure, whatever it is, it has to be completely confidential. It's got to be locked under, if it's a paper form, it's got to be in a locked cabinet, has to be in a locked room. If it's on a computer, it's got to be covered with passwords. And only people who have absolute right and necessary to view the information will view it. When data gets distributed, when you go to a conference or you write in an article and you report it, you should not have any identifying information at all. In fact, when you use statistics, there's no way to break that down unless you give ID numbers, but you should scrub any ID numbers so that you cannot identify a particular person. This may sound kind of silly for stuff that is not really important, but if you have health information and that could actually potentially get somebody fired or get somebody in trouble, let's say you ask them about their boss and they gave you confidential information that could get someone fired and they don't want to disclose that, they don't want to get fired or be a whistleblower, then you have to protect them. There are some health conditions that people don't want others to know about, and it's embarrassing, and they just don't want simply that information out. You have to respect that with health information. And then finally, you must explain what you're doing to people. You have to give them information about the study, usually provide that in the consent form at the very beginning of the study, and then you have to debrief them in the end. You're obligated to debrief a participant, correct any misconceptions about the participant might have had about the research. You have to make sure that you do this at the end of every study. When you're working with animals, okay, instead of the IRB, we have something called the IACUC, and that ethics board is specifically devoted to making sure that animal subjects are being taken care of. So you have to have an acceptable scientific purpose, meaning that you have to be increasing knowledge about behavior, whether it's animals or humans. You have to, have some sort of increase in the understanding of the species you're studying, and it's going to produce results that is going to benefit the health or welfare of people or of the animals. And it must do these things in order for it to be able to be approved. A lot of people think that they have a lot of misconceptions about animal research and think that, oh, okay, well, if you're doing animal research, then all you're doing is torturing animals and that animals aren't being cared for. Well, this is absolutely false. The benefit risk ratio is same with animals. You can't just go and harm animals. You have to have a valid reason, and you have to have a benefit that outweighs the risk to the animal. And whatever you do to them has to be completely humane. So let's say you are studying rats and you want to see if you can figure out a cure for something and you want to test it first in rats. But one thing you have to do is you're looking at their brain, so you have to you have to kill them and cut their head off and actually look at the brain. Well, you actually have to do this in a completely humane way so that the animal doesn't feel anything. The animals that are housed in these research labs have to abide by extremely strict standards, very, very strict standards in care. There has to be a veterinarian who visits the animals regularly and is on the premise for any issues. And they have to be cared for and monitored so that they know that they are, healthy, and that if they have a virus or if they have a problem with them, that they actually are put down very humanely. So this ethics board takes care of this, and also the veterinarian on call has to be available to actually monitor these animals very, very closely. And I've been in a lot of different animal labs, and I can tell you that these animals are very well cared for, by meeting the standards that are in place for this animal research, regardless if it's a mouse or a rat, regardless if it's a rabbit or a non-human primate, whatever it may be, they have very strict standards that they must adhere to in order to do this, to do research with animals. And then finally, this last slide is just a review slide, just to kind of as an outline to help you go back through and study for a quiz if you have it. If you have one, there are some classes that have Chapter 1 quizzes and some that don't. But it's really good to just review because these are concepts in psychology that you really should know about and you should understand. So if you have any questions, please feel free to email me, and I'll be happy to answer them.